Yeah, I'm going to tell a short story today, and I'm going to start first with the acknowledgements, and you saw Trace in the last presentation, but yeah, everything I'm going to show you today is data that he's collected. He's a really talented PhD student in the lab. So I won't delve too deep into an introduction. I think a lot of you will appreciate that kidney disease is bad for muscle. It's a wasting disorder where atrophy and weakness and fatigue are common in these patients, and clearly it's a multifactorial problem. But my lab, I'm a mitochondrial biologist by training, I think, at this point, and much of our focus is on mitochondria. And one of the things that we noticed in the literature is that there's a lot of reports in kidney disease that demonstrate that mitochondrial sort of oxidative phosphorylation is impaired. And so this is a recent example in patients using magnetic resonance spectroscopy or the phosphocreatine recovery kinetics. And yeah, you can appreciate that they see in patients a decrease in the capacity for a calculated ATP production rate if you have bad kidneys. And that same rate was strongly associated with the degree of renal filtration, so a glomerular filtration rate. And what I'm not showing you is it's also associated in this paper with physical performance, so walking speed, strength, those sort of metrics. If you look in the animal literature, there's a lot of studies on how atrophy occurs and the different mechanisms that are driving that response. There are several papers that also show this sort of similar phenotype where they report a decrease in sort of maximal oxygen consumption in mitochondria isolated from muscle of various animal models of kidney disease. But as we sort of scavenged the literature, we came to the conclusion that nobody had really answered this question. So there's clearly a defect, but really a gap in knowledge, which prevents the ability to try to treat or improve this condition, is really a detailed understanding of how the mitochondria are dysfunctional. What are the locations? What are the sites that are impaired in this muscle? And so I set Trace on this path to try to uncover that. So, but before I show you his data, I just want to sort of provide a brief overview of the system that I'm going to show, where we look at the mitochondrial system as a series of batteries that are interconnected, and they transfer potential energy into ATP, which we all know is the currency for our contractile and many other energetic requiring processes in the cell. And so the way we view this is the goal of the mitochondria is we take the fuel that we consume, which will be our first battery, and that fuel, we take electrons off of those complex carbons with these dehydrogenases. So you've all learned the TCA cycle and things like that. That's what these enzymes are for. Those electrons generate a redox charge, NADH or NAD ratio. And then the electron transport system converts that redox potential into the proton motive force. And then that's what proton motive force is what's driving the ATP synthase to create this high energy charge. So lots of ATP in the cell and very low ADP levels. So a key aspect of this is that there are several controllers of this larger reaction where the three controlling nodes can be sort of ATP synthesis and transport, the electron transport system, and then these dehydrogenases. A lot of the conditions, we assume that fuel is not limited. It's only going to be in a starving case that you'll encounter that. So just want you to keep this in the back of your head as I show you the way we've gone about answering this gap in the literature. So first, to show you the model that we've been using is a rodent model in mice where we supplement the diet with adenine, and that will get metabolized in the kidney to 2-hydroxyadenine. And This will cause a lot of pathological events, but one of the things it does is crystallize in the renal tubules. And so it's a nephropathy that develops, and over the long haul, we'll see body weight loss, muscle atrophy, a really diseased kidney, blood urea nitrogen levels, and GFR present like a kidney disease patient. And then, of course, if we look histologically at the kidney, we get nice elements of fibrosis and interstitial inflammation, things that are seen in patients. So first experiment we did that Trace performed was to feed the mice adenine and look at the control over CKD mice. And I'll first explain how we set up this system and what I think is a better way than what has been done in most previous work. So what we do is actually perform sort of a physiological stress test in these mitochondria. And the way we do that is that we use the creatine kinase equilibrium to control the extra mitochondrial ATP and ADP ratio. And that's what the mitochondria are responding to when, for example, we contract. So we think of this as sort of an exercise stress test where we can model the extracellular or extra mitochondrial energy demand at a resting level, and then we can increase it mimicking contractile activity. And this is an example on the y-axis where we're looking at oxygen consumption in the mitochondria, and they're energized by a mixed fuel source of carbohydrates and fats. And we calculate a slope of this relationship as the ability of the mitochondria to adequately respond to increases in energy demand. And much like the maximal decreases in oxygen consumption capacity that are in the literature, we see that this slope is decreased in the mitochondria from CKD muscle. And this is just some modeling of how this reaction works. So as we actually go backwards experimentally, where we start at high energy demand, we titrate in phosphocreatine, and that will decrease the ADP concentration in our respiratory chamber. And so when we see this observation of a decreased slope, there are several mechanisms that could drive that. It could be those dehydrogenases, it could be the electron transport system, it could be the ATP synthase or other substrate transport. So we'll move on. We use the same system, but now we'll analyze a few other parameters of mitochondrial bioenergetics. One of them is to use autofluorescence. So NADH is autofluorescent. We can track that nicely across this change in energy demand. And what you can appreciate from these data is that we see a lower redox charge in CKD mitochondria. Again, and in this case, substrate is not limiting. We're providing that at a saturating concentration. And so the interpretation of that should indicate that those matrix dehydrogenases are probably not working properly. Otherwise, they would generate a normal redox charge. If we look downstream at the proton motive force or the mitochondrial membrane potential, we use a dye called TMRM to do that. And again, we can use it in this context of the exercise sort of stress test. What you can appreciate from these data is that clearly the control in CKD mitochondria are generating similar membrane potentials, suggesting that the electron transport system itself is not dysfunctional, and that the modest decrease in redox potential is not limiting the proton pumps that are in the electron transport system. So now we have some idea, we have some direction that we can go to actually find more information. So to confirm the electron transport system observation here with membrane potential, we use a series of simple biochemical reactions where we provide the electron donor and look at changes in the receptor for complex 1, 2, 3, 4, or 5. What you can appreciate in these data is clearly these individual enzymes, their capacity biochemically is not altered in the CKD condition. So that these data confirm the observation that Trace had in the membrane potential. measurements. So next, we'll go back now to another set of biochemical experiments. So looking at this redox charge being decreased, we have another series of biochemical reactions. There are actually many more than what I'm showing you now. But again, these are individual reactions where we're providing a substrate and looking at the capacity of that enzyme to generate NADH or NADPH in some cases. And what you can appreciate from these data are that several of the matrix dehydrogenases, this is glutamate dehydrogenase, branched chain keto acid dehydrogenase, isocitrate, alpha ketoglutarate, and malic enzyme and pyruvate dehydrogenase, several of these were significantly lower in the kidney disease animals or were very, very close to being significant. Again, good confirmation of this system that we've put in place that can help us diagnose what's wrong with diseased mitochondria. Before I continue that story, I'll just make a quick side note. There's been some evidence or suggestion that ROS might be involved in kidney disease muscle, mitochondrial ROS. And so we did this experiment, which in a manner which a lot of people traditionally do, which is to provide mitochondria maximal substrate in the absence of energy demand. And consistent with those previous studies, we see an increase in hydrogen peroxide production in kidney disease mitochondria. But if we use our system where the mitochondria have even a little bit of energy demand to mimic a resting myofiber, we see that that's not actually the case. There's no difference in the hydrogen peroxide emission or an estimated electron leak from the system. So although we can have an increase in capacity, we don't believe that that's happening physiologically in the resting muscle. So I'll quickly talk about where we're heading and what we're missing. So one of the next questions that came up in regards to the defects in the dehydrogenases was whether or not this is due to a loss of mitochondria. And so Trace has now done several experiments, including some immunoblotting, citrate synthase activity assays, and those biochemical analyses in muscle suggest that the content of mitochondria are proteins are not different. He also recently did a isobaric TMT labeled proteomics analysis of the mitochondria. And what you can appreciate is that very few of the transporters or dehydrogenases involved in generating that redox charge were significantly changed. A lot of the proteins in the mitochondria that were changed in abundance are not related to bioenergetics directly. So we think that this is suggesting that there are some other mechanisms, whether the enzymes are post-translationally modified or if they're directly inhibited by some metabolites that normally would be filtered by the kidney. And I'm going to spend a little bit of time on the latter because we have some evidence there. We're not quite at the stage of uncovering the first question yet. So we did some metabolomics in the muscle of these animals as well. And one of the things that Trace uncovered in those analyses is that there are several metabolites which are classified in the kidney disease literature as uremic toxins. They're metabolites that are normally filtered out by the kidney and excreted in urine. And many of these were significantly correlated. They have a negative correlation with oxidative phosphorylation, the slope of that relationship that I showed you before, suggesting that they may have some impact on mitochondrial energetics.
5 minutes left.
Perfect. Yeah, almost there. So he did some additional in vitro experiments using healthy mitochondria that were isolated from muscle, and then acute exposures to some of these uremic toxins or metabolites that are elevated in kidney disease. in the serum and also in the muscle of these animals. What you can appreciate on the graph on the right is that in most cases there's a dose-dependent impairment in oxidative phosphorylation when you incubate mitochondria with these toxins. And in most cases, this toxicity at the mitochondrial level occurs in concentrations that are much lower than what's in the blood of patients. I think this is the last slide of data, but I'll just highlight that these are ongoing experiments, but some of what he's uncovered is that some of these dehydrogenases are actually inhibited directly by some of these uremic toxins. There's many more that we need to study to really uncover the exact mechanisms, whether they're competitive or non-competitive inhibitors or how they're altering activity, but there's clearly some direct impact of these toxins in mitochondria. And so I'll just summarize and then take any questions. So we've confirmed the other studies that there is an impairment in oxidative phosphorylation in kidney disease muscle, and that it appears that that's derived primarily from decreased dehydrogenase activity without overt changes in content in the system. At least in physiological conditions, we're not able to detect mitochondrial ROS being elevated, suggesting that those might not be directly involved in the pathology. These uremic toxins certainly accumulate inside skeletal muscle when you have renal dysfunction, and they certainly associate with performance and in some cases directly impair mitochondrial enzymes. And I'll stop there and field any questions that you might have.
Great. Thanks a lot, Terence. That was a really nice talk. Do we have any questions right now? I still see Carlo and Paolo's hands up, but I think that's a residual from the previous talk. I've got a question for you while others are thinking. So I just want to get your kind of sense of things. You know, many groups, including mine, have measured mitochondrial ROS generation under those kind of state 2 conditions where you don't have ADP, and so respiration is pretty low. Do you think that has any relevance for resting muscle, or is that just wishful thinking?
That's a debate we have all the time. I don't know. There's never an in vivo condition where there's no energy demand in a myofiber, right? So it's hard to understand what those capacity measurements, and we've used them in the past before as well. It's hard to understand if that relates to even just a resting myofiber. And clearly in the data I just showed, you can see that it's clearly different depending on how we position the mitochondria in those assays. And yeah, I think that question is out there for sure.
Okay, thanks. I see Dario Coletti's got a.
Well, yes, thanks. If you have time, I have an open question for the mitochondria crowd of people. I wonder if this approach of the mitochondrial transfer. I was wondering if any of your conditions you have ever tried to play with it with mitochondria to throw them on either the isolated muscle fibers or, well, for the kidney disease model in vivo. I have no idea how that can be done, but if you have ever even thought about using this approach and see.
Like injecting mitochondria, that's what you mean?
Yeah, basically.
Yeah, I think, I know that literature, I see those studies published. It's hard for me to understand how they survive the high extracellular calcium levels. So I know there's been beneficial reports in the literature, but they might be related to, other signaling that occurs when these proteins or even mitochondrial DNA are sensed. But it's hard for me to understand how they could survive the millimolar levels of extracellular calcium when you just kind of inject them into a tissue.